Hi guys, Professor Ruggieri here again for the Selective and Differential Media Lab. So I wanted to show you basically from the start, you have this package, right, that says refrigerate on arrival. This has been in my fridge. I took it out when I started the water activity and the pH lab. So they've been sitting out for about a half an hour, right? I'm gonna open these up and they should be stored upside down, which is how we want them, right? And they have the label right on there. So there's McConkey, and it says note flecks of dark pigment may occur in the medium. That's okay. Okay, we also have mannitol salt agar or MSA over here. And we have the eosine methylene blue or EMB. So before I actually start the inoculation, I'm gonna take this protective film off of them. And then on the bottom, I'm gonna draw thirds, like a little peace sign, okay? Because you're gonna actually inoculate each of these plates with 3 different species of bacteria. The Staph epidermidis in one, the E. coli in another, and the microluteus in another. If you do not have all these different types of bacteria, you can get in touch with me and I can give you alternatives, or I can upload pictures of the results that I get and you can use those in your lab report. Okay, so I'm gonna undo the packaging on these and draw my little peace sign into thirds on the bottom and write, okay, my initials, the date, and then since there's already a label, I don't need to write MSA or EMB or any of that, but I'll put the species. So I'll put E. coli, M. luteus, Staph epi. Hi guys, so I apologize, this video is gonna be merged from multiple segments, but one thing I wanted to point out is there's a lot of moisture on these. And if you're using a Sharpie, even if you're using a wax pencil, I had to wipe the condensation off the underside of the plate so I could write on it. And you'll see how I labeled it. It says S. epi, E. coli, or M. luteus, my initials and the date. And one thing I'll show you is when I was taking that wrapping off of the mannitol salt agar, I cracked the top. So this is something that happens, right? I'm gonna put a piece of tape on it when I take it downstairs to store it. And I feel like every time I record, there's sirens. But I disregard these, right? These are the inoculated broths from the pH lab. I have these 2, my E. coli and my staph broth. And then over in my refrigerator, I have my M. luteus. Now, ideally, you would want it to sit out at room temperature, right? And it looks clear, but that's because I haven't agitated it. So I'm gonna tighten the lid and then see how it's super full of bacteria. So I'm gonna take this, okay, bring it over here. Now I have all 3 of the cultures that I need to inoculate these plates. Again, if you don't have one of these 3 types of bacteria, consult with me and I'll give you some options as to what you can do. Okay, keep in mind that selective and differential media will favor the growth of certain types of cultures versus another, certain species. So for example, mannitol salt agar tends to favor the growth of gram-positive staphylococci that tolerate salt. So for example, Staphylococcus epidermidis, S. epi, is a species found regularly on the skin. The skin happens to have a very high salt concentration, okay? So we're gonna take a look at all of these. For example, E. coli will turn EMB, eosine methylene blue, a certain color. So I'm gonna inoculate. I'll do 1 plate and then you guys can see how it's done for all 3, okay?